Hello class. So today we're going to cover transcriptional control. And today's lecture is broken into two parts like most of the others. And really the first part is just sort of sit back, take in all the information, and really have just sort of a good understanding of how things work. And then in the second part is really where some of the nitty-gritty details that I want you to know will come into play. So for just a little background, proper control of transcription, it's really one of the most important parts of cell biology. And it controls just about everything. And if you want to sort of take the 10,000-foot view of this, consider for a moment the development of a fertilized egg in a mammal, and how it's just basically a single cell where you're getting DNA from both the mother and the father, and how that actually leads to development of you with all of your different cell types and all of these cells doing different functions. And it does that with basically only about 25,000 genes and 1,800 transcription factors. So just sort of really let that sink in for a little bit and sort of keep that in the back of your head through everything we're going to discuss today and next Thursday. And so one concept, though, that I really want you guys to understand is this concept of activation versus derepression. Okay, when you see a gene expression, its regulation can happen via a number of different ways. Okay, and activation and repression and activation and derepression is just some things that I really want you guys to sort of leave here with. Okay. So there's different types of cells in our body, and we all know that, right? Anyone who's taken systems physiology or anything like that. And here's just sort of an example of a neuron versus a lymphocyte. And think about the different genes that are needed for neuronal development and neuronal function versus what's needed in the bloodstream for, fighting mounting an immune response against an invading, organism. So just sort of keep that in the back of your head that, the complexity of any given cell in the body actually is resulting from a different transcriptional program. Okay? And we all know it comes from our DNA, right? You can take an adult frog and you can take basically skin cells from a frog and culture them in a dish. And then you can take an unfertilized egg and destroy all the DNA with UV light. And then you can actually inject the nucleus from a skin cell, and that frog can then develop into a normal tadpole and then eventually into a frog. Right, and we've sort of all familiar with these cloning experiments. You can do them in plants where you can take a section of a carrot, get the proliferating cells out, then you have a single cell, and then you can reprogram it to basically become an embryo and eventually then a full carrot. And we all have heard of Dolly the sheep, and you can do it in cows too, where you can take epithelial cells from an oviduct or from a cow in general, and then you can remove all the DNA from an unfertilized egg. You can put a cell next to it and fuse those cells, and then you have sort of a reconstructed zygote, which will then develop into an embryo that can be implanted into a cow and would eventually give birth to a calf. Okay, and so ultimately in all of this, it comes down to a timed transcriptional network, right, that alters the gene expression, especially when you're going from, a single cell to an embryo. And I just really want you to sort of appreciate the huge transcriptional program that's going on here. And we have very little of it figured out at this point in time, although great strides have sort of been made in the last 10 years. Now, the way you can measure expression differences in a cell is there's a number of different ways. A really common way that was used sort of in the late 1990s to, basically 2010 or so was this experiment called a microarray. Okay, and it works basically by hybridization. And so what you can do is isolate RNA from a cell. You can label the cells with different colors, Psi3 or Psi5. And then you can hybridize those RNAs to DNA probes that are contained on a slide. Okay, and so here up top are a whole bunch of different tissues and cell types. And going along the y-axis here are all of the 25,000 different genes that exist in our genome. Okay, and so if you see the genes that are going across here are green, that means the expression level is high relative to the population, versus red, which means that the gene expression is low relative to the population. Okay, and so you can see here, if you just look at the prostate, there's a bunch of genes right here that are green and highly expressed in the prostate. And if you go over and compare that to the liver, a lot of those same genes are downregulated and don't express as high. Same thing here, you have these genes down here in the liver that are highly expressed, but not really expressed in the prostate or in breast tissue. And so you can sort of look this over. Each one of these represents a different gene, and so you can sort of see the lines that are going across, and then you have all the different cell types in duplicate running here. And like I said, this was a really popular experiment in the late 1990s into about 2010, but it's since been replaced by this technology called RNA-seq, where you can actually go into a cell and sequence all of the RNAs, and based on the number of sequence reads that you get, you can tell the abundance of a specific RNA transcript. And here's sort of an example of that. Okay, so here you have up top the beta-actin gene. The yellow is the introns, and the red is the exons. And so if you were to isolate RNA from all of these different cell types, embryonic stem cell, liver cell, muscle cell, blood cells, skin cells, lung cells, and you were to sequence the RNAs that were present in those different cells, you can see here how it sort of maps to the genome, okay? And so it's not surprising that in every single one of these cell types, the beta-actin gene is actually expressed. But if you look at tyrosine aminotransferase gene in those same cell types, you'll see that it's pretty much only expressed in the liver cells. And so here you can see the red is the intron, and these reads are actually mapping to the red is the exons, the yellow is the introns, and you can see here that the reads are pretty much matching the exons, and you can see where the areas are actually spliced out. And so once again, this gene's only expressed in the liver. You don't really see it in the other cell types, and so this becomes a very sort of informative way to do this. And one of the neat things with this is you can actually sequence the whole exome and look for potential disease-causing mutations, or you can do the whole genome to potentially find mutations that might be in regulatory regions of the genome. Okay, but ultimately all these differences in gene expressions, they correlate to differences in protein expression. Okay, so here is a 2D gel electrophoresis. And the proteins are stained in red and blue. And if you look at the proteins expressed in a human brain versus a human liver, you can see that the actual patterns of these proteins are different, right? You have this group of proteins that are over here, and you don't see them in the human brain. And likewise, you have some proteins here that you don't really see in the human liver. Okay, so ultimately differences in RNAs lead to differences in proteins, and that gets back to the differences in the transcriptional program that happens between cell types during differentiation. There's also other steps to regulation. There's, of course, transcriptional control, but then there can be RNA processing control, right? You can make different spliced variants of different RNAs that may have different functions or similar functions in different, in the different cell types. And you can also then have regulated RNA transport and localization in the cytosol. And then once you have the messenger RNA, that messenger RNA can actually, its stability can be controlled by either making it so the RNA gets degraded, so there's not a lot of it around. Or you can also have translational control, which will control the amount of protein a given messenger RNA will produce. Okay, and then once you have the protein, there's even further control. You can control the protein activity with post-translational modifications, or you can actually remove the protein through ubiquitination and turnover by the proteasome, which we talked about last time. But if we focus in just on the transcriptional control, we need to go back and sort of look at DNA, okay? Because the regulatory proteins actually associate with the DNA, and oftentimes they associate with the major groove, okay? So it turns out that an alpha helix can fit perfectly in this major groove, and the amino acid side chains can interact with the bases in DNA, right? And that's how you sort of get transcriptional control. And part of it has to do with the chemistry of the double helix. So if you look down the helix, you can see that there's actually H-bond donors and H-bond acceptors and methyl groups, and these can exist in the major groove here, depending on the base pair. This is a GC base pair, an AT base pair, a CG base pair, and a TA base pair. And so these the exposure of these in the major groove, and sometimes even in the minor groove, actually provides nice binding sites for amino acids, so that transcription factors can come in and associate with the DNA in a sequence-specific manner. Okay, and that's key there, in a sequence-specific manner. And so if we look here, this is just another graphical representation of what this slide shows, where you can sort of see you have the H-bond acceptor, an H-bond donor, and just a hydrogen atom, or a methyl group is shown in yellow here, and depending on the sequence will sort of dictate what types of amino acids and what type of transcription factors will like to interact with that DNA. And the same thing happens with the minor groove, and oftentimes you can get amino acids in loops of domains to interact with the minor groove. Here's just a slide showing the different consensus DNA binding sequences of the different regulatory proteins in a whole bunch of different organisms like bacteria, yeast, Drosophila, and mammals. Okay, so you guys have all probably heard about the LAC repressor, LAC-O. This is the LAC operator sequence, and the LAC repressor can actually bind here. In yeast, a famous transcription factor is GAL4, and this is the DNA sequence that it binds to. In Drosophila, we'll see in the next part of the section, there's two transcription factors, Kruppel and bicoid, and here are their consensus sequences. And then here's some sort of famous transcription factors in mammalian cells. P53 is a tumor suppressor, and this is its consensus sequence down here. And so to just give you an idea of how the different amino acids interact with the DNA bases, here you can see a DNA binding protein with an arginine coming off, and you can see that it's forming these hydrogen bonds with the with adenine base. And typically a transcription factor will have 10 to 20 interactions in total, which basically gives it pretty high affinity to bind to a specific sequence of DNAs. Okay. And it's also the DNA folds will sort of help this, and oftentimes, like I said, you'll see alpha helices binding to the major groove. And so here's a sort of famous transcription factor motif, helix turn helix, where you have a helix and then a loop that makes the turn, and then another helix, and this can fit right into the major groove. And so here's just some examples of some transcription factors that use the helix turn helix, the trip repressor, the lambda CRO, the lambda repressor fragment, and the cap fragment. And if you can sort of see that the helix and the helix turn helix, when these things sort of dimerize, they can actually fit perfectly into two major grooves to give it that much more sort of specificity and binding. And that can actually lead to these things called inverted repeats in our DNA, where you'll have the consensus sequence sometimes inverted, sometimes in tandem, that is basically on the DNA. And so in the case with lambda CRO, the consensus sequence is AACAC, and then it's inverted here, so it's CACAA. And the lambda CRO can sort of sit down here and form hydrogen bonds and other interactions with the DNA sequence. There's also another family of transcription factors called homeodomain proteins. These are oftentimes very important in development. And once again, here you have 3 helices, a helix, another helix, and then a third helix there. And here you can see how this helix is fitting perfectly into the major groove with some of its interacting side chains like serine, arginine, and asparagine, and another arginine over here off one of the loop domains can interact downstream. And so these homeodomain proteins are very important in Hox gene regulation, which is involved in a lot of development in developmental programming. Okay. You guys have all probably seen zinc fingers. This happens to be a 2-cis, 2-his liganded zinc finger. So here you can see the zinc, the central zinc. You have 2 cysteines connecting to the zinc and 2 histidines. And then you have your alpha helix that can actually fit in the major groove. Okay. And so here's just an example where these zinc fingers can sort of be tandemly clustered together. And you have this one protein, and then you can sort of see here where you get the interactions between the arginines in the first zinc finger, histidine and arg in the second zinc finger, and 2 args in the third zinc finger are interacting with all of these guanosines in the DNA. Okay. And the more of these domains, the greater the specificity, because the more DNA sequence that it will actually cover and interact with. Okay. And so here's a different zinc finger. This is a 4-cis liganded zinc in it, and you can see here where the zinc finger, the alpha helix in the zinc finger just fits perfectly into the major groove. Occasionally you can have beta sheet binding, where you have 2 beta sheets sort of coming together, and these can actually then fit perfectly also into the major groove of DNA. And finally, you can have sort of loops where you can get the interactions. So this is P53, one of the sort of famous tumor suppressor genes, and not only do you have amino acids coming off one of the central alpha helices in the protein, but then you also have arginines in loop domains and lysines in the loop domains that can also then interact with the DNA. So another type of DNA binding domain in transcription factors is a leucine zipper, and this actually is a coiled domain where you get 2 sort of alpha helices in a coiled coil that can sort of fit down into the DNA almost like a scissors. And these can do kind of great things because you can actually mix and match them relatively simply. Okay, so they can homodimerize, in which case you'll have a direct repeat, but then there's also multiple forms of these, and then so you could have a different one which will bind a different DNA sequence and a different repeat, and then you can mix and match those repeats throughout. You might have instances where these sequences are mixed and matched throughout the genome, where the heterodimers of these actually function, where one helix is binding one DNA sequence and another helix is binding a different DNA sequence. And once again, many different types of transcription factors can actually heterodimerize, and you can also get homeodomain heterodimers, where the different where the different alpha helices are binding to different sequences on DNAs. And then you can have single proteins with combinatorial domains. Okay, so if you look here, you have a homeodomain and then a POU-specific domain in a different part of the protein, and they're connected by a long loop. And this gives just the ability to bind different DNA sequences in sort of a specific manner. A pair of my favorite transcription factors are the helix-loop-helix proteins, and here they actually have a helix, and then they have a little loop, and then another helix, and so the major transcription factors that control the circadian clock are actually helix-loop-helix proteins, and these oftentimes will heterodimerize and homodimerize to give different things. And one of the neat things about the helix-loop-helix proteins is they have inhibitors. Okay, so here's your typical helix-loop-helix binding to an inverted repeat, but then there's inhibitors of helix-loop-helixes that actually don't have the DNA binding domain. Okay, their helix-loop-helix is sort of cut off, and so this can actually associate with the helix-loop-helix that binds DNA and prevent it from associating with DNA. And what's interesting is that there's a specific types of brain tumors called glioblastomas that have these ID proteins, ID1, ID2, ID3, and ID4. And these are helix-loop-helix proteins that actually don't bind DNA. And so if you have a mutation in any of these, oftentimes you get a misregulation of neuronal gene expression and basically the formation of brain tumors or glioblastoma. Okay. Now, one of the common amino acids that are oftentimes in transcription factors is arginine. And one of the great things about arginine is that it can have this sort of shared electron distribution for it to basically allow for this great hydrogen bonding to occur in the arginine. And it's very favorable binding with guanosine. And if you go back a couple slides, you'll see how the arginines in the zinc fingers were tended to interact with guanosine. Now here's just an example of 6 different zinc finger transcription factors and how the different arginines and and histidines and asparagines and other amino acids on the zinc fingers interact with DNA. So this is 5' to 3' DNA, and then 5' to 3' anti-parallel, and you can see where these different zinc fingers are associating with the different bases in this DNA sequence. And so once again, this stuff is sort of for your information because we're really going to get into the nitty-gritty, and I hope the big take-home message here are there's a whole bunch of different types of transcription factors with different DNA binding domains, and the amino acids on these DNA binding domains will actually associate with specific DNA sequences, which then can lead to transcriptional activation. And that will become very important as we go on to part B in the next section. Okay, so I will be back in just a moment